afternoon, and thanks everyone for staying. So I think the most fascinating machines on this planet are ATP synthesis, so I'm going to tell you all about them, and they are involved in biological energy conversion. And back soon. Okay, here we go. So, but first let's start with the most fundamental question of biology. What is life? And I think the most basic answer is that it creates order from disorder and then proliferates. And as you all know, this costs energy. And natural sources of energy are sunlight and any form of gradients. So something doesn't quite work. Okay, but then, so this energy needs to be converted into a usable, storable, and transportable form. So it is speculated that the earliest forms of life have evolved around black smokers at the bottom of the oceans. These exhale liquid, hot, acidic broth that, when encapsulated by a membrane, form a very simple battery with an electrochemical proton gradient across the membrane. So it seems that life has stuck to using proton gradients as energy sources to form the universal energy carrier, ATP, or adenosine triphosphate. And a more modern invention is photosynthesis, which uses photosynthetic reaction centers to harness the energy of light from the sun, build up proton gradients, and synthesize ATP. Now, photosynthesis produces oxygen and high energy nutrients that enter the food chain, and so it ends up in mitochondria, which are organelles in every eukaryotic cell, and they also called the powerhouses of the cell. So the inner membranes of the mitochondria are densely packed with respiratory enzymes, and these use breakdown products from nutrients and produce proton gradients across the inner mitochondrial membrane, and these again are then converted into the universal biological fuel, ATP. So now let's have a look at ATP. It consists of an adenosine base attached to 3 phosphate groups. And the last phosphate can be split off in an energy-producing reaction. So this is a small combustion and gives the power stroke in most energy-requiring biological reactions. So this is like the fuel for our cars, but it is fully reversible and renewable. So in a reverse reaction, the third phosphate can be fused back and reform ATP. So the processes that synthesize ATP are remarkably similar to the processes in a hydroelectric power plant, where the potential energy stored in the water is released by letting the water flow downhill through these pipes that contain turbines. And the turbines contain magnets, and the rotating magnetic field induces an electrical current. So with biological energy conversion, you have proton gradients produced either by photosynthesis or respiration. So the proton gradient in this case goes up and induces a rotation in the turbine of this molecular machine. The torque is transmitted by a central shaft to the catalytic centers. And so ADP and inorganic phosphate come in, and the conformational changes induced by the rotation then fuse the ADP and the ADP together to form ATP, which is then released. And per one full rotation, this produces 3 ATPs. Now, the first near-atomic resolution structure came from this yeast, F1C10 complex, which revealed that yeast ATP synthase has 10 plates in the transmembrane turbine. And each plate translocates one proton through the membrane. Now, this triggered a whole avalanche of reports, and people studied these turbines from different organisms. It turns out that different organisms have different numbers of plates in their turbines, and so they translocate a different number of protons per ATP synthesized. Now, this influences the proton to ATP ratio or the gearing ratio of these machines. So if you look at this green machine here on the right, it uses 15 protons to synthesize 3 ATPs. So if you compare that to gear or riding your bike, you have to pedal very hard to make a short distance. So you use up a lot of protons, but you can go uphill. Whereas in the eukaryotic or mammalian case here, bovine ATP synthase uses only 8 protons to synthesize 3 ATPs, so that would be on a higher gear, so it needs a steeper gradient, and it would be easier to reverse. Now, nature uses various forms of rotary ATPases with different functions, and eukaryotes have 2 rotary ATPases, the F-type ATPase, which resides in mitochondria and synthesizes ATP, and there's an evolutionary related complex, the V-type ATPase, that hydrolyzes ATP, so it works in reverse, and it can pump protons across membranes to build up proton gradients. Now, eubacteria or archaea have usually only one complex that is bifunctional and can operate in either direction, depending on the environment. And so this is controlled mostly by the blades and the turbines, but also by the so-called peripheral stalks. So it is easy to distinguish these complexes. You just count the numbers of peripheral stalks. So F-type ATP synthases have 1. The A or V-type or bacterial V-type ATPases have 2, and eukaryotic V-type ATPases have 3 peripheral stalks, and this all contributes to regulation. Now, because thermophilic bacteria produce very stable proteins, we've used Thermus thermophilus A-V-type ATPase and have focused on this very stable complex that is stable and functional in isolation. So in a collaboration with Carol Robinson and Oxford, we have established mass spectrometry, and we confirmed that this A-V-type ATPase has 2 peripheral stalks. We confirmed the number of plates in the membrane and the weight of the intact molecular machine, which is 660 kilodaltons. But most importantly, Carol's group found that different types of rotary ATPases bind specific lipids to the turbines. So that was a surprise because these lipids are not the predominant lipid species in the respective membranes. So we think that the lipids have a similar function to engine oil in man-made engines. They act as sealants to prevent proton leaks through the membrane. Now, meanwhile, my group determined the structure of a peripheral stalk of this Thermus thermophilus AV-type ATPase. It showed an elongated structure that we could dock into the lower resolution electron microscopy density of the intact enzyme. And we determined 2 different crystal structures, and to our surprise, they showed slightly different structures. So the longer part here shows a bend, if you can see that from far. So these seemingly stable scaffold proteins are, in fact, quite flexible, and we could confirm that by normal mode analysis on a computer. So now when we put this together, it actually starts making sense. So this animation is still fictional. Of course, we don't have structures of all states for this movie, but it should be close. And so I think the flexibility of the peripheral stalks has become meaningful because it allows the peripheral stalks to follow the wobble of the catalytic head group. And if you follow it through, you can will notice that the concerted wobble will prevent the break and reformation of chemical bonds, and that makes them really efficient, and they are close to 100% efficient molecular machines. But even more importantly, we think that they are important for crosstalk, because we think that this structurally still not well resolved transmembrane subunit here takes part in this concerted synchronized movement, and we think that the peripheral stalks contribute to crosstalk, so that the catalytic sites will know what they do, and also what the entrance to the proton channel on the other side, about 25 nanometers away, will do. So if anyone here is interested in motorbikes or engines, you will probably already start to speculate that this is exactly the function of a pushrod. So we think that the peripheral stalks are actually molecular pushrods, and so we wrote a little bit of a fun review, but I think this is serious, so molecular machines have much in common with man-made machines and vice versa, and by now I think I need a degree in engineering. So then we compared, so I said these are thermophilic enzymes, so we compared the peripheral stalks and the dynamics of it with F-type ATP synthase, and so although the sequence and structure is quite dissimilar, the dynamics of F-type ATPase peripheral stalks is similar. So remarkably, electron cryotomography has revealed that F-type ATP synthesis in mitochondria shapes the cristae of mitochondria by forming V-shaped rows of dimers along the cristae. And if you compare mitochondria isolated from different organisms, this is not cooperating. Hopefully just take a second. Here. I hope you still follow. We were at the crevices of cristae from different mitochondria that have different angles in between the F-type ATP synthesis, and this seems to correlate with the numbers of blades in the turbines. So this is just very speculative, still needs to be confirmed, and so we think that the dimerization of F-type ATP synthesis to shape cristae actually supercharges the proton gradient, and so just where it is needed. And so we have trillions of these molecular machines in our bodies that turn over about our own body weight of ATP every day. And so for the biologists, it is speculated that this is the location of the mitochondrial transition pore that triggers controlled cell death. And it was shown by electron cryotomography that these dimers come apart during aging, so they might lead to us losing energy while we age. And it is also likely related to disease. So with this, I would like to thank all my collaborators and co-workers, mostly Alistair Stewart and Lawrence Lee, who are in the audience, and without whom this work wouldn't have been possible. And I would like to thank the ARC, NHMRC, and Victor Chang for founding, and the Academy for the honor to be elected. And thanks for your attention.
Thank you, Daniela, for a fascinating talk. Are there any questions or comments? Tony.
Brilliant talk. Thank you very much, and fascinating graphics. I understand the energy conversion in chemical energy, but what's the mechanical energy got to do with it? What's the mechanics? Well, I don't understand the role of mechanics in your machines.
Of the rotation itself. So that is translated into a movement of conformational changes in the active site of the soluble part of ATP synthase. So it requires energy to get the amino acids that are required to synthesize ATP from ADP and inorganic phosphate. And so this conformational changes, or the energy comes from the rotation, and that is driven by protons. Does that make it any clearer? Not really. We can have afterwards.
That's after later on discussion point. And Ross, please.
Okay. In the theory of heat engines, reversibility gives you the most efficient designs. Is it the same in molecular motors?
I've heard, yes, it is. So, yes. But how do I answer? Yeah, the answer is simply, I've discussed this before, I know it, and it seems to be the case. Yeah, the more reversible, the more efficient they are.
Roger?
Could you bring me up to date, because of my age? Do you still believe that mitochondrial DNA is exclusively maternally inherited? Because the mitochondrial DNA in the sperm midpiece, which controls the beating of the sperm's tail and hence allows fertilization, that is immediately destroyed once the sperm have entered the egg. It's boom, blown up.
Okay. Well,
and,
I suppose
so it's always seemed to be amazing that here's mitochondrial DNA with no paternal inheritance. Is that still true?
I suppose so. I would use the excuse here that I've got a degree in mineralogy, and then I stopped at X-ray crystallography. So this is not really my field, but yeah, that's what I learned too, that mitochondrial DNA is maternally inherited. Is there anyone in the audience who can?
Okay, maybe there's a very good discussion point probably you can have. Maybe last question, we'll have Graham to ask the last question. Yeah.
I was just wondering about, in terms of the numbers of protons, is it 14 for higher plants and 14 protons per ATP for higher plants?
For plants, yes.
Yeah,
What's that?
Yeah, and are they all the same?
Well, there's spirulina is 15. So, and I don't think, well, there aren't that many species that have been analyzed by mass spectrometry or structural biology, so it's still open whether there are maybe 13 or 15. So there's a lot of variation.
Okay, on that note, could you please join me to thank Daniela for a fascinating talk?